Welcome, everyone. Today we are going to be taking a look at a Step 1 histology crash course that I've put together. My name's Keaton Erickson, and let's go ahead and get started. So I wanted to start off with a few tips and tricks in general regarding histology, basic histology, even attacking pathology slides. First and foremost, when we are considering normal histology and trying to learn that and trying to implant that into our brains, we have to recognize that most of our organs are organized into fairly organized and recognizable layers. And they're very similar across different systems in our bodies. So if you consider the layers of the GI system, right, we have our mucosa, our submucosa, our muscularis externa, and then our serosa or adventitia on the outside. That's very comparable to the wall of the ureter or the wall of the trachea. They're very similarly oriented, where we have our epithelial layer, and then we have some submucosal layer for blood vessels and connective tissue, and then we have a muscular layer oftentimes, and then we have an outside connective tissue layer. And that is pretty well conserved across different systems within our body. Like I said, the GI system, the respiratory system, the urogenital system, that's fairly well preserved across all of those systems. So if you put some time into memorizing, let's say, the GI system and the layers associated with that, that will transfer to other systems as well and make that learning pretty easy. And then second of all, as far as step one is concerned, a lot of the questions regarding specifically normal histology are going to rely on those structure function relationships. So why is this epithelium present here? What do these types of cells do in this sort of circumstance or in this part of the body? Smooth muscle is not under voluntary control. Those types of structure function relationships, identifying something on a histology slide, and then being able to transfer that onto a physiological, response, etc. Those structure function relationships are really the bedrock of histology and how it's very easily testable when it comes to the step one exam. And then I really like histology because it's fairly predictable. Like I said in the first point, right, the recognizable layers across organ systems is pretty well conserved. And thus, we just have to know a few specific things about each different organ. So what type of epithelium is present in a certain portion of the body. But even that we can begin to predict. So memorizing some structure function relationships as it relates to specific epithelium types, that can get you a long way. So for example, stratified squamous epithelium is really good at protecting the body from outside invaders, as well as friction forces. Whereas columnar and even simple squamous epithelium is not the best suited for that. So in portions of our body that are going to encounter a great deal of friction forces, as well as the outside of our body trying to protect ourselves from foreign invaders or antigens, it makes sense to have a stratified squamous there. But it doesn't make sense to have a stratified squamous epithelium lining the GI system because it's not well adapted for absorption or secretion like we see in that GI system. So thinking about those structure function relationships can get you to predict a lot of different useful knowledge and testable knowledge as it relates to histology. So if you were going to devote some time to a histologic, you know, topic to cram for step one, I would advise to try to memorize the epithelium types present in the different types of tissues. That is very high yield and can be very easily testable on step one. All right. And then finally, how Step One and UWorld and other Q banks like to test histology concepts is trying to see if you know what's normal and then apply that to a pathology. So I have a few examples here. Respiratory distress syndrome of the infant, that is due to, not well developed lungs yet. So we don't have enough surfactant produced by those type 2 pneumocytes. And to test that, a step 1 question might show you a picture of normal lung tissue, normal adult lung tissue, and then ask you which of the cells is implicated in that pathology. Similarly, Barrett's esophagus, that is the metaplasia of that stratified squamous epithelium to a columnar type epithelium like we see in the stomach and the rest of the GI system, but found within the esophagus. So Barrett's esophagus is a pathologic adaptation to repeated chemical exposure. And so recognizing that if we see columnar type epithelium within the esophagus, well, that's not normal, right? So they might ask something about that. And then similarly, vaginal adenosis, that is the persistence of a columnar type epithelium found within the vagina, where we normally see a stratified squamous epithelium, right? So knowing the normal, recognizing what pathology is associated with those normals, that is going to get you a very far way as it pertains to step 1. Okay, so those are my intro tips and tricks. Yeah, epithelium types for sure, the corollaries to pathologies, and then the structure function relationships are going to take you far as histology concepts are concerned. Okay, so my agenda for today is to pretty quickly walk through all of these 7 systems. We're going to start with our skin and then end with our cardiovascular system. I'm going to try to boil down what I think is particularly helpful and testable as it pertains to step 1. All right, so let's go ahead and start with our skin histology. So skin histology, as it relates to step 1, is pretty simple. Okay, all we have to know is basically the normal structure of skin. There's a lot of pathologies that have representative histology images, but as far as normal histology is concerned, you really just need to know the 5 layers of the epidermis and then recognize that thick skin and thin skin look a little different from one another. So as far as the 5 layers, we have our stratum corneum out here on the very much outside. We have our stratum lucidum, which is this almost translucent is how I like to think about it. You can't really see individual cells. They all sort of blend together. That layer is going to be our second outermost. And then our third outermost, our middle layer, is going to be our stratum granulosum. That's going to be our middle layer. That is going to be where our keratinocytes begin to have some granules, hence the name granulosum, and appear hyperchromatic and more basophilic. And then we have our stratum spinosum, which is the sea of keratinocytes that have intercellular bridges. Yeah, that is going to be our second most inner layer. And then we have our stratum basale, which is our basement layer portion of our epidermis. This is where our melanocytes are housed, and our stem cells that are responsible for producing new keratinocytes. So they're also going to be found at the stratum basale. So remember the 5 layers, remember their names: corneum, lucidum, granulosum, spinosum, and basale. And then also remember what different cell types are found within each. So the corneum is going to be made up of keratinocytes that have since lost their nucleus. Lucidum, same thing, beginning, it's that transition phase from nucleated keratinocytes to non-nucleated keratinocytes. That is basically just keratin up here. 3, granulosum, still just keratinocytes. 4, the spinosum. Here we start to see a few different cells. some dendritic cells, and then keratinocytes as well. And then our stratum basale, that is going to be where we see those melanocytes with a halo around it, and then definitely some pigment that the melanocytes have produced and spread to those keratinocytes at the basale layer. All right. So pretty simple as far as the normal histology. The epidermis is the most highly testable. So I would commit to memory the 5 layers of skin for sure. And then I wanted to contrast thick skin, like we saw before, with very well-defined layers of our epidermis, and then contrast that to our thin skin section here, where we see a very well-defined keratinizing layer, okay, very well defined, but we don't see much of the stratum granulosum or the stratum lucidum at all. We have a pretty thin layer of stratum spinosum, and then we have that stratum basale layer. So thick skin has a lot of stratum corneum. That corneum layer is very well formed, and that is extremely well adapted to resist friction forces. So thinking of that structure function relationship, where might you find thick skin? Well, in areas where you are going to come into a lot of friction forces. So specifically the palms of our hand and then the soles of our feet is where you find thick skin. Thin skin is found pretty much everywhere else in the body. So if you were to take a sample, like a biopsy of skin from, say, the abdomen, this is probably what it would look like. A very well-defined keratinizing layer, stratum spinosum, and then stratum basale layer. Okay. So just the differences, try to recognize those because it could be a testable question. Just where in the body would this have been taken from? Something along those lines. All right, and then a lower yield topic as far as step 1's concerned is the location and types of glands that we see in our skin samples. So we have 3 different types of glands. We have sebaceous glands, eccrine glands, and apocrine glands. And then I have the relevant material summarized here. Sebaceous glands are going to be located directly next to a hair follicle, and the contents of the sebaceous gland are going to be deposited onto that hair follicle as well. So close in proximity, and it drains directly to the hair follicle. And then the type of secretion, and I have seen questions like this before, as far as sebaceous glands, is going to be that holocrine type of secretion. The holocrine type is where it basically the whole cell degranulates to release that type. So all of it is released onto the hair follicle, and that cell ends up dying in the process. Transitioning to the apocrine and eccrine glands, these are going to be located in the deep dermis, right at that dermal hypodermal junction. They follow a merocrine type secretion system, but they'd have different draining points. So apocrine glands are going to be draining onto the hair follicle, similarly to our sebaceous glands, whereas eccrine glands, these are going to drain directly to the skin surface. All right. So eccrine glands drain to the surface, apocrine glands to the hair follicle. So let's take a look at some of these. So we have 3 different types of glands here. Just to orient ourselves, this is going to be near our epidermis up here, and then moving further down is going to be our hypodermis underneath these glands. We can start to see a bit of adipose tissue creeping up, and that's where our hypodermis starts. So up in the up next to a hair follicle, we are going to see our sebaceous glands. They appear pretty pale staining in nature, and they're often clustered together in close proximity. Move this out of the way. This structure up here with a lot of basophilic staining keratinocytes, this is going to be a hair follicle structure. So this sebaceous gland is located in close proximity to that hair follicle, and it will drain via a holocrine secretion system into this hair follicle right here. So sebaceous glands are located closer to the epidermis, whereas our apocrine glands and our eccrine glands, apocrine over here on the left, eccrine over here on the right, these are going to be our deep dermal sweat glands. So these widely distended glands here, they have a lot of room within the tubules, within the glandular structures themselves. These are going to be our apocrine sweat glands. And then our eccrine sweat glands have this appearance of having almost two different structures found within, but it's really these pale staining glandular structures that are going to be our eccrine glands. And then these basophilic tubule structures here are actually going to be the ducts through which they release. So as a refresher, these apocrine glands over here on the left, they are going to drain most likely onto this hair follicle right up here, whereas the eccrine glands are going to drain via these ducts all the way to the skin surface. Another point to be made is that these apocrine glands are just found in select portions of our bodies, and they play no role in heat regulation whatsoever. Whereas our eccrine glands, these are the sweat glands that you think of when you think about sweating in response to heat. So eccrine glands are going to be our heat regulators. Apocrines are under the control of our sympathetic nervous system and respond to emotional stimuli as well. So let's take a closer look at those glands here. As we can see on the left here, our apocrine glands have a lot of luminal space in them. That's often what I sort of view as, mnemonic for apocrine glands is going to be these widely distended glands deep in the dermis that will often be next to eccrine glands here. So our eccrine glands have pale staining cells, much like our sebaceous glands, not as much lumen to be seen, nowhere near our apocrine glands here. And then they are often in proximity to these duct structures that appear dark staining and are found within, interspersed in between our glandular acini here. All right, that wraps up skin. As far as a pathology correlate, I wanted to throw some of these in here just so that we can see both normal and abnormal in the same slideshow. So the pathology that I've chosen for skin is a melanoma. Our melanocytes, I think I mentioned just briefly, are going to be found within that stratum basale layer in normal skin tissue, and they appear to have like a halo around a really dark nucleus. So that's going to be representative of a melanocyte, but here we can see we have far too many. For the most part, they are staying in that stratum basale layer and not entering into any deeper structures. So I believe this was termed a melanoma in situ because it has not invaded the basement membrane yet. But we do see some characteristic features of melanoma where we have a melanocyte that has actually jumped up into the stratum spinosum. That is pretty characteristic of some melanoma subtypes. And then I wanted to ask a question here just to test what we've learned so far. Where in the body might we have taken this particular biopsy from? Give you a second to think about that. All right, so how do we go about answering a question like this? We have to identify, is this thick skin or is this thin skin? If it's thin skin, well, almost anywhere in the body, except where we find thick skin, is going to be thin skin. But this particular sample has a very well-established stratum corneum, a little bit of a stratum lucidum, and then a pretty recognizable stratum granulosum here. So with the very thick stratum corneum layer and a well-recognizable stratum granulosum, we would term this thick skin, which basically limits us to the palms of our hands or the soles of our feet. Alrighty, let's move on to respiratory histology. There is plenty to know about respiratory histology as a whole, starting in the nasal cavity, going all the way down into the larynx, the trachea, bronchi, bronchioles. But as far as this presentation today, I'm going to focus on some higher yield questions. First of all, what type of epithelium are we going to see in the respiratory system? And then I'll talk about cartilage and cilia as well. So as far as the epithelium, this is one of those oddball cases where it looks a certain way, but it's actually not that. So we see a lot of nuclei present here, and they seem to be at different levels within the epithelial layer. So our epithelial layer is all the way from up here down to here, and it goes across. This is going to be our basement membrane. So these appear to be very tall and skinny cells, and there's a lot of nuclei. So one might want to say that this is a stratified epithelium, but this is actually one of those weird cases where this is a pseudo-stratified columnar epithelium. All of the cells here extend down to the basement membrane, but some might not go all the way up. So each cell within this epithelial layer is in contact with this basement membrane down at the bottom, but they might not go all the way up to the top. And then another structure we're seeing up here is these frilly little structures up on the luminal surface. These are going to be our cilia. So our respiratory epithelium from our nasal cavity all the way down to our bronchi is going to be this pseudo-stratified columnar epithelium. And then we also are likely to see goblet cells. This is likely a goblet cell. Those are going to be our pale staining structures within that epithelial layer. So epithelium in our respiratory system, for most of it, is going to be that pseudo-stratified columnar epithelium with cilia and goblet cells. Yep, so it extends all the way down through our bronchi. When we get to our bronchioles, that's where we start to see a transition to a cuboidal epithelium. And then as we know in our alveolar spaces, those need to be very thin, very well adapted for gas exchange. So that's where we're going to see a simple squamous epithelium. So pseudostratified columnar epithelium with cilia all the way down to through our bronchi. When we get into our bronchioles, that's where we start to see cuboidal epithelium. And then finally, down in the alveolar space, that's where we start to see our simple squamous, well adapted for gas exchange. Okay, let's transition to talking about cartilage. In the trachea, we have several rings of cartilaginous material. Hyaline cartilage rings that they function to keep our trachea patent and open so that air is easily moved through that structure. And those cartilage pieces start to become a bit more irregular, but they're present all the way to the end of the bronchi. The absence of cartilage on histology slides should indicate that we are not looking at a bronchus anymore, but instead we are looking at a bronchiole. So if we don't see cartilage, we know we're past the point of bronchi. So cartilage is found all the way from the trachea down to the end of our bronchi, but not found in bronchioles at all. Now, cilia, like we said, these are our cilia up here. These cilia are found all the way until the end of the respiratory bronchioles. They begin to be a bit fewer in number, but cilia are found at the level of the respiratory bronchioles, no further though. They're not found in the alveolar space. And that makes sense, right? If something gets all the way down into our alveolar space, we want structures there to help remove that debris from our alveolar space to aid in gas exchange. So cilia are there as a protective, almost cleaning mechanism to move things up and out of our respiratory system. Alrighty, so let's take a look at some of those things. This is a sample from the trachea. Here we have those cartilaginous rings that I was talking about. We have our luminal space here. This is where gas is, or air is moving up and down. And then we have our epithelial layer that is going to be that respiratory epithelium, that pseudostratified columnar epithelium with cilia and goblet cells. What we see here is an example of a bronchus. We can tell that it's a bronchus because we're still seeing columnar type epithelium, which we see all the way through our bronchi. We start to see cuboidal epithelium down in our bronchioles. So that's one element of identifying this as a bronchus. But really what I can hang my hat on as identifying this as a bronchus as opposed to a bronchiole is the presence of these circular structures here. These are going to be examples of irregular plates of cartilage, which are seen all the way through our bronchi. And I will repeat this, they are not seen in bronchioles. So if we start to see cartilage within our tissue sample here, we know we're at the level of a bronchus and not a bronchiole. Alrighty, and let's move further down our respiratory system, down all the way to our gas exchange area, our alveolar space. And there's plenty to identify here. So most of our structure here is going to be made up of a simple squamous epithelium. These cells are termed type 1 pneumocytes. So we have an example here. We have an example here, out here. And then in the actual walls of our alveoli are going to be coursing capillary structures, which makes sense because that's needed for efficient gas exchange. And we can see those capillary structures as filled with these red blood cells in the actual wall of our alveoli. So here's a good example of a type 1 pneumocyte with capillaries running directly next to it. And then often found at the corners, the intersections of our alveoli. So here's an alveolus, here's another one, and here's another one. At the corner, at the intersection of these three, we see this more uniquely staining cell here. It has a nucleus that has a irregularly staining pattern with some granules out in the cytoplasm. Here's another great example here. So an irregularly staining nucleus with white granules found in the cytoplasm. These are going to be our type 2 pneumocytes. And these serve a couple of functions within our respiratory system. The most clinically relevant one is going to be that they are responsible for surfactant production. So these would be the cells implicated in a respiratory distress syndrome of the infant, because these cells have not matured enough to produce adequate amounts of surfactant quite yet. So type 2 pneumocytes are responsible for producing surfactant down in the alveolar space, but they also act as a sort of stem cell of sorts, where if there's damage to the alveolar space, these type 2 pneumocytes will differentiate further and then replace those damaged type 1 pneumocytes. And so they will differentiate into the type 1 simple squamous epithelium present down in our alveolar space. All right, and that pretty much sums things up. We don't see great examples of a alveolar macrophage here, but those would be found within the space here. And then sometimes you can see phagocyte host material. Yeah, so that wraps up our alveolar space respiratory histology. And then speaking of those alveolar macrophages with contents found within them, these cells, these strange staining cells here, are found within an alveolus that has a bunch of debris found in it, likely to be fluid. This is a lung sample taken from the patient with left heart failure. The pathology here, heart failure is impaired forward flow of blood due to compromised contractility or filling of the left heart. But here, what we see on histology are going to be these quote unquote heart failure cells. These are going to be alveolar macrophages filled with hemosiderin. Okay, so hemosiderin filled alveolar macrophages that should start to make you think of heart failure in general, but more specifically, backup of flow into the lungs. Okay, yeah. So, like I said here, hemosiderin-laden alveolar macrophages, and these cells are termed heart failure cells because of the corollary there. Alrighty, let's move right along to our GI histology. I want to start with just a more in-depth discussion of the layers because they are important as far as GI histology is concerned. Basically, we have 4 different layers of the GI tube from esophagus down all the way to the rectum. This first layer is termed the mucosa, and that is further divided into our epithelial layer, a directly underlying lamina propria, and then a small layer of muscularis mucosae. So we're going to see smooth muscle just at the transition point from mucosa to submucosa. Our submucosa is primarily composed of connective tissue, and it will often have vessels associated with it. And then in some portions, we might even see glandular structures in the submucosa. Our muscularis external layer, with the exception of the esophagus, it is going to be a smooth muscle layer, often thick for our GI system, that is responsible for moving the contents of our GI system along and distally. And then finally, we have a connective tissue layer on the very outside, termed adventitia or serosa, depending on where you are intraperitoneally or retroperitoneally. So yeah, how I've broken up our GI system here as far as this review is concerned is basically just a picture of the organ that we're talking about, and then what I think you should know about it for STEP. So we're going to start at our esophagus. The epithelium is going to be a stratified squamous epithelium without keratinization. So we don't see any of the flaking off pieces at the luminal edge. We see a stratified epithelium, and then the outermost cells here are going to be that flat squamous cell type. So this is termed a wet or without keratinizing stratified squamous epithelial layer. And the interesting part about the esophagus, besides the epithelial layer, is the contents of that muscularis external layer. So when we take a closer look at that muscle layer, depending on where we are within the long tube known as the esophagus, we will see different types of muscle composition within that muscularis external layer. So up in the upper 1/3, so near our neck and within our neck, the esophagus muscularis external layer is made up of skeletal muscle only. Okay, skeletal muscle only. As a refresher, for skeletal muscle histology, we're going to see peripheral nuclei. We're going to see cross striations. Yeah, I mean, that pretty much wraps it up. If you see peripheral nuclei, that should make you think of skeletal muscle. Okay, and they're often bigger cells. And then we get into the middle 1/3, shown here, where we see a mix of the skeletal muscle type, and then we have smooth muscle as well. So as we transition from the upper 1/3 down closer to the stomach, we're going to see an increase in the smooth muscle composition and a decrease in the skeletal muscle composition. So in the middle 1/3, we're going to see a mixed proportion of skeletal muscle fibers as well as smooth muscle fibers. And then in the lower 1/3, we are just going to see smooth muscle. There will be no more skeletal muscle as we approach the stomach there. All right, that is our esophagus. Let's move on to our stomach. So if we recall from anatomy, our stomach is actually going to be made up of four regions. we have our cardia, which is directly near the lower esophageal sphincter. It's the opening of the esophagus into the stomach. That is going to be neighbored by the fundus, which extends superiorly just a little bit, and then the body, which makes up the most area of our stomach. And then finally, as we transition from stomach into our next organ, the small intestine, we are going to see that structure, that region called the pylorus. So yeah, cardia and pylorus are very similarly, histologically speaking, and the fundus and body are also very similar, histologically speaking. So in the cardia and pylorus, we see these glandular structures shown at the right here. They are going to be monotonous glands. They're moderately sized monotonous glands, monotonous meaning that all of these cells appear to be the same from lumen all the way into the deep portions of our glands. They often have a pale staining appearance, and that's because these glands are responsible for producing high amounts and great amounts of mucus material to prevent the neighboring structures from being damaged by the strong acid of our stomach. So the cardia and the pylorus have glandular structures that have a pretty pale staining appearance, and that's because they are producing mucus-like material to protect neighboring organs. We contrast that to our fundus or body histology. These glands are very long, so they're very well-developed glands, and they appear to have several different types of cells found within them. And that's true because there are several different types of cells found within these fundal and body gastric glands that are responsible for producing different types of materials. So up here in the lumen, we first encounter our gastric glands, and we have this portion up here. I like to refer to it as the upper 1/3 portion of our gastric glands. These are going to be our mucous neck cells. So these serve a similar purpose to the glands we just saw in the pylorus and cardia. They are producing mucus-like material, but these help to protect the actual body and fundus from the other components of these gastric glands. So mucus neck cells produce mucus to help protect against the harsh environment. The middle 1/3 are these very pale staining, almost fried egg cells. These are our parietal cells. These are going to be responsible for producing the hydrochloric acid as well as intrinsic factor to help in B12 absorption. So parietal cells are found within the middle 1/3 of our body and fundal gastric glands. They appear as these pale staining, almost circular fried egg structures, and they are responsible for gastric acid and intrinsic factor production. And then finally, down at the deep portions of our glands, we have these very bipolar staining cells, where the inner portions are staining this bright eosinophilic color, and the nuclei located peripherally are very basophilic staining. So these are our chief cells, and they are responsible for producing pepsinogen, which of course is converted to pepsin in the lumen that helps break down proteins. So as a summary here, up in the upper 1/3 of our gastric body glands, we have our mucus neck cells that produce exactly that, mucus. In the middle 1/3 of our gastric body glands, we have our pale staining fried egg appearing cells, termed parietal cells. These are responsible for producing gastric acid as well as intrinsic factor. And then finally, the lower 1/3 of our gastric glands, these are our chief cells, as well as a few other cell types that I didn't mention here, that are responsible for producing a zymogen called pepsinogen, which is further converted into pepsin to help break down proteins. So mucous neck cells, parietal cells, chief cells, as we go deeper into the glands. Alrighty, let's move on to our next organ, which is the small intestine. The small intestine first starts off with our duodenum, transitions into our jejunum, and then finally our ileum before we get to our large intestine. So our duodenum is the shortest portion of our small intestine, and it is histologically different from the other portions of the small intestine because of the presence of submucosal glands. These are called Brunner's glands. Nowhere else in the small intestine are you going to find glandular structures in the submucosa. So just as a refresher, our mucosa contains our epithelial layer, an associated lamina propria, and then a little bit of muscularis mucosae. Okay. And then we have our glandular structures found within our submucosa, which is specific for duodenum. Nowhere else in our intestines are we going to see glandular structures found within the submucosa. So these Brunner glands are diagnostic for a small intestine biopsy or sample. And then finally, we have our muscularis external layer and then a bit of connective tissue for our serosa or adventitia. All right, so this is our duodenum. Remember, submucosal glands for duodenum. Next, we get into our jejunum. Here we see what I like to think is our classic small intestine histology. We have villi that are extending upwards, as well as neighboring crypts, termed the crypts of Lieberkühn, that house our stem cells that are dividing and replenishing these villi epithelial cells as they slough off. So just to repeat that, we have villi extending outwards into the actual lumen of our small intestine, of our jejunum. And then we have crypts that extend further deep into the wall that act as a safe place for our stem cells, for our epithelial tissues in our GI system. And then this here is our submucosa, which in the jejunum is really just occupied by vessels, loose connective tissue, and then some lymphatic vessels as well. And then we have our muscularis externa down at the bottom here. So as far as a small intestine biopsy or sample, if we don't see glands in the submucosa, then we know we're not in the duodenum. No duodenum if there's no submucosal glands. If there is no if there's nothing else found within the submucosa, then we're likely looking at a jejunum sample. And then let's contrast that to our last portion of our small intestine. This is going to be our ileum. Similarly, we're seeing these villi extending into the lumen, and then we also see some crypts extending deeper into the walls. We are not seeing submucosal glands, but instead we're seeing a different structure that is diagnostic for the ileum. The ileum contains these highly developed, pretty specific, pretty recognizable structures of immune cells termed Peyer's patches. So Peyer's patches are found within the submucosa of our ileum, and they help to protect against the highly bacterial contents of our large intestine. So the ileum is the Peyer's patches are diagnostic for an ileal biopsy or sample, and they are found within the submucosa of the ileum wall. So just to reiterate, just as a summary, let's go back. Duodenum has these submucosal glands, not found anywhere else within our small intestine. They also have villi and crypts. The jejunum lacks anything special in the submucosa. So it really is just occupied by connective tissue, vessels, and lymphatic vessels. But here we're also seeing villi and crypts. And then our ileum, we see villi and crypts again, but the unique feature of the ileum is going to be these collections of inflammatory and immune cells termed Peyer's patches, and also secondarily termed mucosa-associated lymphoid tissue that are sitting within the wall, within the submucosa of our ileum. Alrighty, let's move on to our large intestine now. Our large intestine differs from our small intestine histology because instead of seeing those villi that we were just looking at, all we see in the large intestine, the large intestine, are going to be these crypt structures. So nothing extending upwards. We have a flat layer here, and then everything extends down into the wall as opposed to extending up into the lumen. So our large intestine, to repeat, has no villi, but has highly developed crypts in the wall. In the wall of the crypts, we see that these crypts are actually staining very pale. There's not a lot of color to these cells at all, except for the nuclei at the base. That's because these are very highly occupied by goblet cells, producing a lot of mucus, producing protective enzymes, and releasing those into the contents of this organ. And that makes sense because of the highly bacterial contents of the large intestine lumen. So many goblet cells, only crypts, and no villi. That's what we see with the large intestine. The submucosa is pretty sparse. It's really just connective tissue and vessels. And then we have our muscularis externa, and then a connective tissue layer on the outside. So it's a pretty standard organization as far as GI is concerned. But the really important part is that there's no villi in the large intestine, and then they have a great deal of goblet cells found within those glandular structures. Alrighty, and then we're transitioning to the last portion of our GI system as far as the alimentary canal is concerned, and that is the transition from rectum into anus. So the rectum, we see portions here. We see no villi. We see some glandular structures that are extending into the wall. So these are going to be our crypts and a lot of goblet cells found there. Submucosa, and then we have this odd structure that's not found in normal portions of the large intestine. This is a sphincter, as well as this number 2 out here. These are 2 sphincters associated with our anus. But before I get there, I want to talk about the transition from epithelium types to epithelium types. So we have a simple columnar epithelium found within our rectum. And then as we go along, we start to see this transition into a very well-defined, stratified epithelium. This is going to be that same epithelium that we found in the esophagus. It's going to be a wet or non-keratinizing stratified squamous epithelium. And then as soon as it's not pictured in this sample here, but if we were to look at an external sample of the anus, we'd likely start to see keratinization on that sample as well. So internal structures are not going to be keratinizing. External structures, you could expect to see some keratinization there. So important to note that the transition from rectum to anus shows a transition from simple columnar epithelium to a stratified squamous non-keratinizing epithelium. And then let's talk about these structures here. These are anal sphincters. Just geographically speaking, we can make an intuitive guess that number 1 is going to be our internal anal sphincter, and then 2 is going to be that external anal sphincter. But if I were to zoom in on these 2 structures here, we'd likely see a difference in the makeup of the muscle structures there. So number 1 is going to be that internal anal sphincter, which, if we recall from physiology, is not under voluntary control, while number 2, that external anal sphincter, is under voluntary control. So based on that knowledge and our structure-function relationships, we would expect to see that number 1, our internal anal sphincter, is composed of smooth muscle, as opposed to number 2, which is under voluntary control, would be made up of skeletal muscle, which is what we see if we were to zoom in. So number 1, our internal anal sphincter is composed of smooth muscle fibers, whereas number 2 is composed of skeletal muscle, which allows us to control the defecation process. Alrighty. Now we're getting into some of our secondary GI organs. So let's start off with the liver. If you recall, liver histology is composed of repeating hexagonal lobules. So there's tons of them. They're all neighboring one another. An actual histology slide is very rare to see a perfectly well-defined hexagonal structure, but I've approximated one on this slide here. So the structure found in a liver lobule is going to be a central hepatic vein. So we see some blood cells found within that hepatic vein there. And then hepatocytes that are stacked and pointing, they seem to radiate out from that central hepatic vein. And then we have corners that are made up of portal triads. So we're not seeing, like I said, perfectly defined liver lobule here, but we can start to see some structures here, here, here. And if I were to zoom in here, we would see remnants of 3 vessels. We'd see a branch of the hepatic portal vein. We'd see a branch of the hepatic artery. And then finally, we'd see a branch of a bile duct that is leading actually out of the liver inferiorly towards the duodenum. Whereas the flow of material into the liver and through these portal triads is going to be superiorly, and it will flow through these sinusoids found within these hepatic lobules towards the hepatic vein here, and then these will further drain back up into the inferior cava and empty into the right atrium. So just to repeat, we have branches of our portal triad vessels found at the corners of our hepatic lobules. In the middle, we have a central hepatic vein. We have sinusoids that are extending in between stacks of neighboring hepatocytes. Blood flow will go towards the hepatic vein here. And then within those sinusoids, we're actually going to see some resident macrophages. These are termed Kupffer cells, and they are macrophages that are specifically resident macrophages found within the liver sinusoids. So that's just a fact to put away. The cells found within the hepatic globules that act as macrophages are termed Kupffer cells. One portion of histology that I haven't really addressed on this slide is the presence of stellate or itocells. These are also cells that are found within the liver parenchyma. They act as reservoirs for vitamin A. And then additionally, they are responsible for responding to inflammatory signals and producing and initiating fibrosis. So stellate or itocells, hard to identify on a histologic sample, but I don't want you to just forget about those cells. So those cells act as vitamin A storage, and then also respond to inflammatory signals to initiate the fibrosis process. All right, that's liver. Let's move on to pancreas. Histology of the pancreas is pretty straightforward. Most of the entire organ is made up of these secretory acini structures. And by acini, I just mean a small collection of cells oriented in a circle fashion, almost like a cul-de-sac, with ductal structures draining each one of them. So they will each drain into small ductules that drain into a further until it reaches the main pancreatic duct. So most of the entire organ is termed an exocrine pancreas because these secretory acini are secreting digestive enzymes, which then get emptied into the duodenum to help break down food and help absorb those nutrients. And the contents of these secretory acini never get released into the bloodstream. They're just released via ducts into the duodenum itself, and that's why it's termed an exocrine portion. But the endocrine portion of the pancreas are contained within these isolated islets of Langerhans. These are going to be composed of beta cells, alpha cells, delta cells, and are responsible for producing insulin, glucagon, somatostatin, and some other hormones that are less clinically relevant. But these are obviously the structures in our pancreas that are implicated in processes like diabetes. So they're sort of the structures in the pancreas that don't look like the rest of them. That doesn't look like the rest of the organ itself. All of this looks pretty monotonous. They're pretty identical cells, but they're just arranged in little cul-de-sac circle structures. But then we have these circles of these nests of cells that don't look like the rest. And those are termed the islets of Langerhans, and are the endocrine portion of our pancreas. Like I said, they're responsible for housing the cells that produce insulin, glucagon, and somatostatin and some other hormones as well. So that's the pancreas. Alrighty, we're coming to the end of our GI histology here. I wanted to include a pretty common pathology correlate that you could likely be tested on test day for step 1, and that is celiac disease. Of course, there's a lot more to know about celiac disease than what I've listed here, but as far as the histology is concerned, what you would likely see is it would be termed a duodenal biopsy that doesn't really look like duodenum at all. We would expect to see on normal duodenal histology the presence of villi, some crypts, and those submucosal glands. But on a celiac patient, we are likely to see blunting of the villi. We are likely to see crypt hyperplasia, so very well extensive crypts that extend into the wall as opposed to into the lumen. And then finally, if we look closely enough, you might be able to see intraepithelial lymphocytes. Now, those are sort of the classic features of celiac disease, and what you need to know for step 1 histology's sake. Of course, there's a lot more to know, like the associations with HLA types, the DQ2 and DQ8, as well as the diagnosis or the confirmatory portions by measuring IgG or IgA, anti-gliadin, anti-TTG, and anti-endomysium antibodies. But as far as histology is concerned, the classic features of celiac disease are blunting of the villi, crypt hyperplasia, and intraepithelial lymphocytes. Alrighty. So let's now move on to our immune system histology. We're going to start looking at components of the blood and then proceed to secondary and primary lymphoid organs. So red blood cells. You are likely to see a peripheral blood smear or 5 or 6 on step 1. Likely there's going to be a pathology on those red blood cell peripheral smears, of which I have not really listed here. But this is a normal peripheral smear with pretty standard red blood cells. They are biconcave in shape, in 3-dimensional shape, and that appears on histology as a pretty well-defined sphere with a central white spot. And also remember, in the maturation process of erythrocytes, we ditch that nucleus as those cells mature. So we are going to see anucleate cells with a central pallor because of that biconcave disc, and pretty eosinophilic staining on the edges. I would try to familiarize yourself with what normal red blood cells look like, because you might be asked to identify microcytic or hypochromic or macrocytic red blood cells, all of which have a differential diagnosis associated with them. In the case of microcytic hypochromic, your first thought should probably be iron deficiency anemia, of which many pathologies can cause. But as far as normal histology, I would try to familiarize yourself with how much eosinophilic material is there, how big should that central pallor be, and then, yeah, if you're looking at a pathologic blood smear, some tips I would say are to compare red blood cell sizes to one another. They should all be relatively similar to one another, like we're seeing on this slide here. All of the central pallors should be similar, and they should appear like we're seeing here, with pretty significant portions of the peripheral eosinophilic material present. And then, yeah, obviously look for abnormal red blood cell shapes. Here's one, but because it's just one cell here, I wouldn't think too much of it already. So as far as studying for step 1 goes, I would definitely try to put some time into memorizing or understanding the hematology labs and their associated conditions. And then also there's a list in first aid of all the pathologic erythrocyte shapes and their associated conditions. So that definitely is easily testable material. So try to put some time in there. All right, now we're just going to go through the peripheral white blood cells found in a peripheral blood smear and what their morphologies look like. I'm not going to dive too much into their structure-function relationships or where we see increases or decreases, but just focusing on morphology to help you out with identification. So the 2 cells that we're seeing here, these are going to be examples of neutrophils. They have pale, sort of hard to make out granules in the cytoplasm, and then the nucleus is highly lobulated and pretty irregular in shape. So it has a segmented nucleus that's pretty recognizable as far as its shape is concerned. It's irregular, it's lobulated, it's segmented, and then it has those pale granules out in the cytoplasm. Moving right along, here is another granular or granulocyte example. Here we see a cell that has a lobulated, segmented nucleus, but the color of the granules are incredibly different than that which we just saw in neutrophils. These are very striking, very eosinophilic granules found within the cytoplasm. So this is an example of an eosinophil. So compare and contrast. These granules are hard to make out and pale in the cytoplasm. This is a granulocyte with very striking eosinophilic pink granules in the cytoplasm with similar nuclear architecture, segmented and lobulated. All right, and then here's a very rare cell to see on a peripheral blood smear. It's hard to even make out the nucleus, but we can see one portion of it here and then another bit here. So again, this would be a segmented, lobulated nuclear structure. But the most striking feature of this cell is that it also has cytoplasmic granules, but these are incredibly dark and purple staining. So this would be termed a basophil because of the basophilic granules found within the cytoplasm. All right, moving along, these 2 cells here are our next white blood cell that we'll talk about. They basically just look like floating nuclei, and that's because the nuclear architecture of these cells occupies almost the entire volume of these leukocytes. These are termed lymphocytes. So these white blood cells really just look like basophilic nuclear architecture with a very little bit of cytoplasmic material surrounding it in between the cell membrane and the nuclear membrane. So these are basically just floating nuclei, and they are lymphocytes. All right, and then our final leukocyte that we'll take a look at here is a monocyte. So they are cells that are bigger than red blood cells. they have a nuclear architecture that almost looks like a C, and it is often eccentric, meaning off to one side, and it has significantly more cytoplasmic material than the cells we were just looking at. Very little cytoplasmic material. Almost the entire cell is occupied by that nucleus. Here, the nucleus is large and takes up a big portion of the cell, but the cytoplasm clearly takes up a considerable portion of the volume of the cell. So C-shaped nucleus that is often eccentric and has not insignificant cytoplasmic material. So that would be termed a monocyte. Alrighty, let's transition to our lymph node histology. I'm not going to spend too much time actually talking about the portions of the histology of lymph nodes, but as far as it relates to pathologies and just basic immunology concepts, they're incredibly high yield to know. So out in the cortex here, out in the outer portion of our lymph node is termed our cortex, and we can see a lot of germinal centers. Okay, we see a lot of these, we see a lot of active germinal centers out in the cortex, indicative of an active lymph node that is going through an inflammatory process. So these germinal centers found within these structures out in the cortex, these are going to be where we primarily locate B cells, our B lymphocytes, and this is where they are undergoing, undergoing immunology processes to help specify their specific antibodies. So out in the cortex is where we find germinal centers and specifically B lymphocytes. Here in what's termed the paracortex is where we find a great deal of T lymphocytes to help initiate antibody production and facilitate the interaction between T cells and B cells. And then we have our medulla here. This is where lymph fluid is going to flow from the cortex into the medulla, and then finally into our efferent vessel, exiting and then flowing back up into our either another lymph node or back into our blood. Let's see, what else have I missed? Yeah, so I mean that pretty much covers what you need to know as far as lymph node histology. We have our germinal centers out on the outer cortex of our lymph nodes. We have our paracortex that is just a little bit underneath, and that's where we find a great deal of T cells. And then we have our medulla here, which is where lymph flows into, collects, and then eventually drains into an efferent lymphatic vessel. One other point I should mention is that lymph flows into the lymph node through portions, breaks in the cortex, and then it will flow past and through all of the cortex and into the medulla and then out. So the vessels that drain into a lymph node are termed afferent vessels. The vessels that drain out of the lymph node are termed a single efferent vessel. All right, and then I just wanted to get your minds thinking as far as a pathologic condition, what areas are we likely to see missing in a patient with hyper IgM syndrome? Think about the pathology there, and then what would we likely see on a lymph node biopsy like this? So that is going to be the absence of those germinal centers out in the cortex. Hyper IgM syndrome results because there is not efficient class switching of those B lymphocytes. And so the absence of class switching would show an absence of those those paler staining portions out in the cortex. Alrighty, let's move along to our bone marrow. As far as You don't really need to know too much about bone marrow for step one as far as the histology or pathology is concerned. But there are a few things that I think that you should be able to do. And that is, first of all, recognize that this is a bone marrow sample. Recognize that these structures here and then within the actual marrow itself are bone fragments that we got from a biopsy. And then another question that could come up pretty easily is to show you a very hypocellular bone marrow sample where the relative abundance of fat is way above normal. And of course, getting down into the specifics of that would maybe require a pathologist level of education to know. But if they show you just adipose tissue in what they call a bone marrow sample, then you should be thinking of processes like myelodysplastic syndromes, like myelofibrosis. If it's completely replaced by adipose or fat tissue, then that is indicative of a pathology. Probably unlikely that you're asked that, but recognize that in a normal bone marrow sample, there is a balance between cellular components as well as fat tissue. So it would be termed hypocellular if fat tissue predominates too much. And then finally, the other component that I would think that could be tested pretty easily is asking what type of cell that this would be within a bone marrow sample, and that would be a megakaryocyte. So megakaryocytes are often multinucleated, very large in comparison to the other cells around it, and are often in close proximity to a vessel of some sort. So this megakaryocyte is in close proximity to two vessels, likely extending its cytoplasmic extensions into the vessel there and releasing platelets into the peripheral blood. Alrighty, move on to our last immunologic organ, and then this is our thymus. As far as the histology, again, I would say it's pretty low yield for you to know too much about the thymic histology. However, what I do think you should know is recognize that it's broken up into a cortex and a medulla. The cortex is extremely basophilic and dark staining, whereas the medulla is more eosinophilic staining and is more centrally located. And then studying more immunologic functions of the thymus, I think that that is high yield. So knowing what portions occur in the cortex and what occurs in the medulla. And then finally, what I think you should know is what does a thymus look like as we increase in age? And that is going to be that this portion here involutes and is replaced by a great deal of adipose tissue. So instead of seeing really robust lobules here, we would likely see fat predominant with a few maybe functioning lobules of thymus. But it incredibly decreases in robustness and volume as we reach adulthood. Okay, let's take a break for pathology correlates to our immune block here. The pathology that I've isolated here is really pathognomonic because of this structure here and this one here. So these are going to be our rods, and they are very classically associated with acute promyelocytic leukemia specifically, but acute myelocytic leukemia more generally. So our rods associate with acute promyelocytic leukemia. That specific pathology is associated with a 1517 translocation. And you can see these either on peripheral blood smear or in a bone marrow sample. And then as far as treatment, all trans retinoic acid is an option here. But really on histology, look if they show you or if they give you a description of a a pretty classic leukemic patient, and then on blood smear, they show these lines out in the cytoplasm, be thinking acute promyelocytic leukemia. Alrighty, let's move on to our GU system. So we're going to start with our kidneys, work our way down, and then we'll take a look at some reproductive organs as well. Kidney histology is surprisingly high yield when it comes to step one. So be able to recognize that this sample here, first of all, comes from a kidney, and then be able to recognize specific portions of this. So how do I know that this is kidney? Well, it's usually because of this structure here. Okay, this is usually what I hang my hat on if they give me a sample and are requiring me to know what organ it's from. That structure is pretty specific. You don't see these marble, I like to think of them as marbles in a field of tubes. You don't see those anywhere else in the body, really. So glomerulus with a Bowman's capsule, that white space on the outside, and then it's surrounded by a great number of tubules that are pretty similar in shape and size, with some associated vessels. So be able to recognize the following on kidney histology: a glomerulus, tubules, and then I'm going to show you another picture out here. This one is going to be our transitional epithelium. So transitional epithelium, I'll show you in a minute, but it appears to be a stratified cuboidal epithelium that when distended will appear as a stratified squamous epithelium. So I will show you a picture when we get to the bladder of a more clearly identifiable transitional epithelium sample, but it is pretty darn specific for the GU system. It's really only found in the ureters and bladder in our body, so that if you see a transitional epithelium, be thinking of the GU system in particular. Yeah, glomerulus, tubules, transitional epithelium. I think that that's, as far as normal histology is concerned, that's what you should concern yourself with. Recognize that this structure is a glomerulus, and it should help you to get to that point if you just see a bunch of tubules surrounding that glomerular structure. And then finally, one note here is that the glomeruli are only found in the kidney cortex. You don't see those in the kidney medulla at all. All you're going to see on histology of the kidney medulla is tubules, and specifically collecting ducts and some loops of Henle. But collecting ducts, loops of Henle, that's all you're going to see in the medulla. If you see a glomerulus, you know you're looking at a cortical biopsy of a kidney. Alrighty, here is a good picture of transitional epithelium. And honestly, as far as histology is concerned for step one, I would really only memorize the fact that transitional epithelium is found in the ureter and bladder. Other than that, the histology is not incredibly important. There are some muscle structures that they could test you on as far as it relates, but I think that that would be a stretch. So memorizing transitional epithelium and associate it with the bladder and ureter, that will get you as far as I think you need to go. So what makes this a transitional epithelium? Well, down here is our basement membrane. So we see that there are many different cells that are stacked upon one another. So that makes it stratified. But when we get to the top, we see these cuboidal cells, which should be a surprise. Stratified cuboidal epithelium does not really exist in the body. So when you see cuboidal cells on top of a stratified squamous, just stratified epithelium, that should make you instinctually choose transitional epithelium. Because why this is transitional, why it's called that, is because in the ureter and bladder, when it becomes distended with urine, this epithelium allows for some stretch, and these cells, when they're stretched, flatten to become a squamous shape. So it becomes a stratified squamous when distended, so it transitions into that epithelium. So a transitional epithelium appears as a stratified squamous epithelium, or excuse me, stratified cuboidal epithelium when not distended, and distended will appear as a stratified squamous epithelium. Alrighty, let's move on to some reproductive organs here. This is a transverse section of the penis. As far as histology is concerned, I would really just try to remember where the fascia and where the corpora cavernosa and corpus spongiosum are located on histology. So I'll identify them here. So number 1 is going to be our tunica albuginea that surrounds our corpora cavernosa. So these two structures are arterial structures, and they become engorged and distended with blood upon an erection. So these are the structures that are filled with blood when a penis is erect. We move on to our corpus spongiosum. This is going to be what surrounds the urethra in the spongy urethra portion of the penis. And then 4 is going to be our buck fascia. This is often talked about in terms of like a penile fracture or like a urethral rupture or damage. So buck fascia is going to be outside of the tunica albuginea, and then the superficial fascia will be even further out than the buck fascia. So tunica albuginea, that's the structure that surrounds the corpora cavernosum, both of them, and then the corpus spongiosum surrounds the urethra itself, and the buck fascia encircles all of it. The superficial fascia will circle all of that as well. All right, moving down into the testis. These are honestly pretty simple histology structures, luckily. There's just a few things to remember, is that the testis is broken up into many seminiferous tubules in which spermatozoa are undergoing division and maturation processes. And there is a significant blood testis barrier to prevent autoimmune attacks, basically, of those developing sperm cells. And that is accomplished by these cells here, termed Sertoli cells. So Sertoli cells are in charge of maintaining that blood testis barrier and are located on the periphery of our seminiferous tubule. What's found in between the seminiferous tubules are termed the interstitium, and more specifically, found within this interstitial tissue are the interstitial cells of Leydig. So the cells of Leydig are the ones producing testosterone and are found in the interstitium in between adjacent seminiferous tubules. So yeah, that pretty much wraps up the testis. And so now we'll switch over to female reproductive organs. So we'll start at the ovary. You could spend probably 3 hours memorizing what each developing follicle looks like at each stage, but I would say that that's relatively low yield for step 1. What I would say is that you should be familiar with the different portions of a developing follicle, and that is that this structure here, this cell in the middle, the big one, that is going to be our developing oocyte. The layer on the innermost side of the developing follicle, that is going to be our granulosal cell layer. And then just external to that is going to be our theca cells. So there's plenty of theca cells on the outside surrounding each developing follicle. But as far as histology is concerned and step 1's concerned, I would know to this level that the oocyte is the big internal cell surrounded by a layer of granulosal cells, and then finally an outermost layer of theca cells. All right, let's turn our attention to the uterus now. There are super important pathology correlates associated with the uterus, specifically adenomyosis or placenta accreta or increta. Those pathologic conditions are really dependent on if you know the layers and the general organization of the uterus. So up here, this developing layer here is going to be our endometrium. So this is the layer that's responsive to a woman's menstrual cycle. And then deeper down, we have a myometrium, or a muscular layer of the uterus. And that this is going to be, responsive to signals to contract in terms of, delivering a pregnancy. So internal most is going to be our endometrium. These glands will adopt various conformations as it relates to how far along in the process, how far along in the menstrual cycle a person is. And then the myometrium, that's going to be the very thick muscular layer of the wall that's going to be responsive to signals related to delivery of a pregnancy. All right, here's our pathology correlate. Sorry for the poor quality of this picture, but it was the most obvious explanation or demonstration of this pathology correlate that I've chosen. The correlate is termed vaginal adenosis. It's a pretty rare condition, but it is a very easily testable condition as it relates to histology. So knowing the normal histology of the vagina is really how this could be tested. So normal histology, normal epithelium type of the vagina is going to be a stratified squamous epithelium with keratinization if it's external, but if it's more internal structures, it will lack that keratinizing layer. Vaginal adenosis, however, is the persistence of columnar epithelium found in the upper 2/3 of the vagina. So if we see columnar epithelium found within a biopsy of the vagina, we know that that's abnormal, because normal epithelial layer there should be stratified squamous epithelium. So persistence of a columnar, a simple columnar epithelium is definitely going to be evidence of a pathology here. And so this is really associated with that DE, that diethylstilbestrol exposure in uterine, while the developing fetus is in the uterus. So yeah, on histology, we see normal stratified squamous epithelium over here. And as we transition, we start to see only one cell layer thick as far as the epithelial layer, and they are taller than they are wide. So that would be our simple columnar epithelium found there. And we really only see adenocarcinoma of the vagina in this particular condition. You can certainly have other types of cancer, but if it reads in a question stem that a patient was diagnosed with adenocarcinoma of the vagina, we should start to think about exposure to this medication here, and the pathophys of persistent columnar epithelium where it's not meant to be in the vagina. Alrighty, briefly, we'll go over a few nervous system histology concepts. Really, I think the main thing to get down as far as nervous system histology is being able to recognize glial cells if they show up. And I think the most testable glial cell is going to be this first one, these oligodendrocytes, appearing very similar to what we saw on skin histology, right? They are a fried egg with a halo around the nucleus cell type. The difference is obviously the context in which these cells are found. So the context of oligodendrocytes is they are primarily going to be found within the white matter. So we're not seeing a lot of neuronal cell bodies around, and that's because oligodendrocytes are, of course, the myelinating central nervous system cells, and so they will be wrapped around the axons of our neurons. So oligodendrocytes are primarily going to be found within the white matter of our nervous system, and they take on this fried egg halo around a nucleus appearance. Okay. Astrocytes, sorry for the image quality here, are a little bit harder to identify just on normal histology, but they have a similar appearance, except they don't really adopt that halo around the nucleus morphology. It's really just a nucleus and then surrounded by neuropil just around it. But the thing that I would hang my hat on is that these cells are found directly next to a neuronal cell body. And like we just said, oligodendrocytes are primarily going to be found within the white matter, so not right by the neuronal cell bodies, whereas astrocytes are. If somebody were to tell you that a stain was used and that these cells lit up, and then asked you what stain it was, GFAP is specific for astrocytes, and can be used in like identification and classification of a brain tumor derived from astrocytes, like a glioblastoma. So GFAP positive cells should make you think of astrocytes. All right, microglial cells. These are going to be also found within both gray and white matter, but they appear as this sort of blunt cigar-shaped nucleus. Yeah, they look different than everything else around them. Most of the other cells have a pretty pale staining nucleus, maybe a halo around the nucleus, whereas microglial cells are just this cigar burnt, blunt-ended shape nucleus. Yeah, and you can see a great deal of them in conditions like encephalitis or meningitis. These cells will respond to infection. They're derivatives of monocytes that are just found within the central nervous system. So microglial cells are immune cells that can increase in number in inflammatory conditions. All right, and then finally, we'll just take a quick look at ependymal cells. These can simplistically be thought of as the epithelial cells of the central nervous system. So these are the cells that are lining our ventricles within our CNS, and they are the ones responsible for producing our CSF. And you'll notice on the outside, on the luminal surface, we see a lot of debris out here, and that's because these cells are ciliated and they also have microvilli. So ependymal cells, you can basically think of them as the epithelial cells of the CNS ventricles. Alrighty, and I'm going to move directly onto cardiovascular system histology. First, we'll take a look at the heart, and then we'll take a look at arteries and veins next. But as far as the histology is concerned, I would say, for the most part, that normal histology for both of these, both arteries, veins, and the heart, are lower yield for step one. They might show you, a pathologic biopsy of a heart, for example, post myocardial infarction, but normal histology is going to be a little bit rarer, I would say. Regardless, to go through it, the heart is broken up into 3 layers, as are the vessels. Internalmost, so the lumen of the heart, is here. We first reach our endocardium. So the endocardium is lined by a simple squamous epithelium, a bit of connective tissue underlying it, and then we have a portion termed the subendocardial layer that is composed of Purkinje cells and other conducting system cell lines and materials as well. So endocardium is our first portion. It's pretty thin. It is a simple squamous epithelium on the internal most layer, some connective tissue underlying it, and then portions of our conducting system of the heart in our subendocardial layer. All right, and then we'll move forward just a little bit to look at our myocardium, which is our next layer. To review, these are made up of cardiac myocytes, which appear a little bit different from skeletal muscle and smooth muscle. So to review those concepts, in cardiac myocytes, we are going to see cross striations Okay, we see those. They appear as faint lines that run across each cell. Specific features for cardiac myocytes are that we see branching. So we see one cell here, and then it branches into a left and right branch. We are likely to see intercalated discs if we look hard enough. Let me see if I can find one. Here's one. We see intercalated discs running across the width of our cardiac myocytes. And these, of course, are allowing for, cation and anion exchange between adjacent muscle fibers. Electrolyte, that's the word I'm looking for. Electrolyte exchange and message communication between adjacent cardiac muscle fibers. And then if we were to see a cross section, we would see centrally located nuclei. So if you see a cross striation, a cross section of cardiac myocytes and the nuclei are located centrally, we know it's not skeletal muscle because those nuclei are located peripherally as opposed to centrally located nuclei in cardiac myocytes. Alrighty, and then finally, we'll take a look at our epicardium, which is our outermost layer of our heart. This is composed of adipose tissue, our great big coronary arteries and veins, and then we have a visceral layer of that pericardium. We're not seeing the parietal layer of our pericardium because it's not contained within this section. But the thing to remember about the epicardium is that the great coronary vessels are going to course through this layer. It has some cushioning due to this adipose tissue on the outside. And then that visceral pericardial layer is attached to the actual epicardium itself. All right, that wraps up heart. Let's now take a look at vessels in general, and then specifically answering the question, is this an artery or is this a vein? So let's start off by identifying our layers of vessels that we're talking about. As far as layers of the vessel, they are termed tunics or tunica. They each have 3 layers. We have a tunica intima, okay, TI, or tunica intima. We have a tunica media, and then we have a tunica adventitia or externa. So tunica intima is going to be composed of that epithelial layer, the simple squamous epithelium, and then a few variable components, usually a connective tissue layer, a basement membrane. And then in arteries, we will see this structure that we're seeing here, this internal elastic lamina, this squiggly line here. That is going to allow for more elasticity and then some rebound effect to accommodate for increasing blood volumes in high pressure systems. Tunica media is really just composed of smooth muscle that is responsive to catecholamines and sympathetic nervous system inputs. And then our tunica externa or adventitia is going to be our connective tissue layer that blends in with the surrounding tissue around it. So similar to the heart, the correlating layer would be the tunica intima is going to be our endocardium. Tunica media is going to be that myocardium. And then the epicardium is going to be our tunica externa or adventitia. Alrighty, so let's look here. We have our lumen here. Now we're trying to answer the question, is this an artery or is this a vein? I think it will probably be more niche and will not show up on STEP. That being said, it's useful knowledge, I think. So our tunica intima is pretty thin in this vessel and not well defined. But then we'll take a look at our tunica media and tunica adventitia. So the tunica media is going to be that muscular layer, and then the tunica adventitia is that connective tissue layer on the outside. So the tunica media, in comparison to the tunica adventitia, will almost always answer this question for you. The ratio of media to adventitia will almost always answer this question for you. If the adventitia is much larger than the media, you're almost certainly looking at a vein. I'll repeat that. If the adventitia, the width of the tunica adventitia, is substantially larger than the tunica media, you're almost certainly looking at a vein. Now, if the media is bigger or about the same size as the adventitia, then you are almost certainly looking at an artery. So let's take a look at a couple examples here. So I've listed this as a vein. So let's try to confirm that by looking at a few characteristics. So one thing I haven't quite talked about yet is the shape of the lumen. So if we see a really irregularly shaped lumen with some spikes and some weird corners being turned, that's going to make me think it's a vein. And then I would try to look for some supporting material also. So this lumen here is going all over the place. It is incredibly irregular. It's filled with blood cells, as you can see, but it is taking some really random curves that you'd not expect in an artery. So this, just based on lumen size and lumen shape, you could think of as a vein. Now, if you were to try to map out the width of the adventitia and media layers, it's a little harder because it's not a super well conserved sample here. But let's take here. So this is the width of the media. And then I would just keep going. And you see that this whole layer out here is the tunica adventitia. So they're pretty comparable to one another. But I would say that there is more adventitia to tunica media. Regardless, this would be a hard sample to necessarily say that because it's not well preserved and you can't really tell where adventitia stops and surrounding connective tissue starts. But we can take a look here though. This is a pretty regularly shaped lumen. It's not perfectly circle, but there's no random corners or turns that would be unexpected. And then let's take a look at the width of the media layer compared to tunica adventitia here. The media is much thicker here as it is compared to this vessel here. In addition, we're also seeing a rather squiggly line right at the beginning of the vessel wall here. And that's indicative of the internal elastic lamina, which is present in arteries, but is not present in veins. So to summarize, the media layer is going to be thicker in arteries. Internal elastic lamina is going to be present in arteries, but not veins. And then also the ratio of media to adventitia is going to be larger in arteries than in veins. My final note is take a look at the lumen shape. If it's highly irregular, like this one, meaning it has turns and corners, you should think that it's most likely going to be a vein and look for some supporting material around it. Alrighty, our final pathology correlate here is going to be an aortic dissection. The pathology here is that a tear in the tunica intima allows blood to enter the wall of the aorta, and it will often collect in between the tunica adventitia and the tunica media layers. On histology, what we see here is we see a collection of blood cells that is most likely coagulated and formed a clot in this false lumen. And we know it's a lumen, or a false lumen, excuse me, because we can see that this layer here is a very clearly defined smooth muscle layer here. And then out here, all we're seeing is connective tissue. So we know that blood should not be here because the normal structure of a wall should go tunica intima, tunica media, and then a connective tissue, tunica adventitia, on the outside. But here we're seeing blood and space in between the tunica media and the tunica adventitia. So we know that this is a dissection because blood and fluids are not supposed to be in between smooth muscle and the connective tissue layers of our vessel wall. And then risk factors, there's plenty of them. There's more than I listed here, but the main ones are hypertension, especially hypertensive emergencies, smoking, trauma is also associated with it, and then some inherited conditions like Marfan syndrome and Ehlers-Danlos syndrome. Alrighty, well, that wraps up what I've prepared. I wish you the best of luck and hope this was helpful. Bye, everyone.